Hi everyone. In this video, I'm going to show you how to take raw mRNA sequencing data and process it in Galaxy, an online web server. Specifically, our goals will be to generate some raw counts, so pretty much the number of reads that were detected for each gene in this experiment, and how to identify differentially expressed genes. These would be genes that are significantly upregulated or downregulated between two or more different samples. So first of all, I'd like to give you an overview of what this process is actually going to look like, since by the time we jump into Galaxy, it's just going to be clicking a lot of buttons and putting names in fields. Okay, so this is what we're actually doing at the granular level today. All right, so first of all, this video assumes that you've already done some mRNA sequencing and gotten the raw data from those experiments. Now, in most cases, the raw data from those experiments will be what are called fastq files. Now, a fastq file is a very large file. It's probably around 2 gigabytes in size because it contains 20 to 30 million reads from an mRNA sequencing experiment. All right, and each one of those reads is going to contain some sort of label, followed by the sequence that was actually read by the sequencer, all right, and then a quality score that indicates how confident we are in each one of these bases that are part of the read. All right, so we've got about 20 million of these different entries in the fastq file. That's a good start, but it's nowhere near a count of how many reads we're getting per gene. Okay. Oh, but first, I also wanted to mention the data that we'll be looking at today is from an mRNA sequencing workflow where we did a paired end read. Now, what that means is that every time a fragment, like this one shown down here, that goes dark blue, light blue, dark blue, every time one of those fragments was sequenced, it was sequenced from one end, that's the forward read, and from the other end, that's the reverse read. Okay? So that's an important thing to know because the tools that are built into Galaxy will ask us if we had single end or paired end reads. So it's good to know that at this point. And anytime you do sequencing, whether by yourself or with a third-party vendor, they will tell you what type of workflow, what type of reads you have. Okay? So all of that is contained in these fastq files. It's just the fact that it's a paired end read means that we actually have 2 fastq files for every sample that we submitted here. We have one fastq that's for these forward reads, and we have another fastq that's for the reverse reads. Okay, so there we go. We've got our fastq data. The first thing we're going to do is we're going to take these sequences and we're going to align them to a reference genome, in this case, the human genome. Specifically, we're going to use the HG38 build. There's a lot of different versions of the human genome out there that are available. This is the most recent one that's available to us, so that's what we're going to use. Okay, but we're going to use this tool HiSat to take those sequences from the fastq data and align them to that HG38 genome. Okay, and that's a very important step in this process because that's what's actually going to correlate each of these reads with a specific spot in the genome, or more importantly, a specific gene in the genome. So you can see over here, an example of the output for HiSat would be something called a SAM or a BAM file, and a typical file like this will have a name for that read, and then it will tell you the position of where that read aligns somewhere in the genome. So here, I believe this is telling us that this read aligns to chromosome 1 at this position right here, at about 10,052,000 bases into it. All right. And then we have the sequence itself and some more quality scores to show you, how confident are we in this alignment. All right. So HiSat takes our mRNA sequencing data and aligns it to a reference genome. And again, you should know what your reference genome would be because you extracted the RNA from these cells. In this case, we extracted them from human cells, so we're using the HG38 human or Homo sapiens reference genome. Okay, just a few more details on this file before we move on. First of all, there are several different options available to you in Galaxy and elsewhere for doing this operation, for aligning fastq files and reads to reference genomes. In this case, we're going to use HiSat because it's one of the preferred tools for mRNA sequencing because it is splice aware, which means it is aware that we are working with mRNA sequencing data, which would contain reads that might have exon-exon junctions. And you need to keep that in mind because when you go to align that type of read to the genome, there might be an intron between those two exons that are joined together in the transcript. And HiSat can account for that. Okay? So that's why we go with HiSat. But even so, there's other splice-aware alignment tools that you could also use. I just wanted to let you know that. The output for HiSat is going to be a BAM file, but it could also be a SAM file. Both of these files tell you the same exact information, which is where these reads align to the genome, but the SAM file is human readable, so you could open that file and actually interpret what is being said there. A BAM file, though, is binary, so it's a bunch of ones and zeros, and that's something that you can't readily read as a human, but it does allow us to compress these files, okay? So a BAM file would be about half the size, or maybe even smaller, than a SAM file, all right? And these files are huge. They're going to be gigabytes large, just like the fastq files. So usually people will save things as BAM files. They'll save their alignments as BAM files to, minimize the footprint on the hard drive that we're taking here, okay? And that's especially important for Galaxy because you have a certain quota in Galaxy. You can only save so many files there, and then they limit you, all right? So we're going to save our alignments as BAM, but SAM is another option that you have. Once we have these alignments, though, we're still not done. We still need to count how many reads are there per gene. All the BAM file is telling you is where all of your reads align to the genome. And for one reason or another, some of those reads might not even be in a gene, okay? They might be in another region, okay? But what we really want to get out of this exercise today is we want to know how many reads or how many sequences do we get for every single gene in the transcriptome, okay? And for that, we use another tool called feature counts. And what feature counts will do is it will go through your BAM files, looking at where all these reads are, and then it will count for every gene in the genome how many reads map to that specific gene. So you can see an example of what that data would look like down here in the bottom right. For this gene right here, this is an NCBI gene ID. So for gene ID 1, we got 2 reads in these control samples, but then 0 reads in the third, 0, 2, and 1 in these transfected cell samples. Okay, so what you're looking at there is literally the number of times that that transcript popped up in these mRNA sequencing reads. Okay, so 2 is a fairly low number. Hundreds are still low. You can see up to thousands or even more, just to give you a little bit of context there. But this is what we really want to get out of that mRNA sequencing data, is these counts. Because once we have those counts, then we can do some interesting things with them. So for example, I'll show you here in this video how to go from these counts, or how to use these counts with a tool called DESeq2 to identify differentially expressed genes. So these would be genes that are expressed at a statistically significant higher level in one sample relative to the other. Okay, so in this case right here, where we have untransfected and transfected cells, DESeq2 will tell us, is this gene right here, this gene ID 1000, is that significantly higher in the transfected cells than it is in the untransfected cells? That's what DESeq2 tells us And that's exactly what we want to get out of this experiment as it happens. All right. So it gives you a p-value over here. And so you could say, I only care about genes that had changes in expression that correlated to p-values that were less than 0.05. And so you can use that to really narrow down your list of genes that might be differentially expressed. The other thing that DESeq gives you is a log2 fold change. So this is a measurement of how much that gene has expressed. All right. So this is telling me here that this gene was 2 to the 7th power lower in the transfected cells than in the control cells, for example. Okay. And together, we use that log2 fold change and p-adjusted value over here to define what a differentially expressed gene actually is. Okay, so this is definitely something that's, up to the individual user, but typically we define a DEG or a DEG as a gene that has a p-adjusted value less than 0.05 and a log2 fold change that is greater than 1, which in other words means that it changed at least twofold its expression level. Okay? So that's how we identify differentially expressed genes in Galaxy, is using this DESeq2 tool. Okay? The other thing we can do with these counts is we can also take these counts and calculate what's called a transcript per million value, or a TPM. And this is a very useful measurement because what it does is it takes these raw counts and it normalizes them according to the length of the corresponding transcript and the total number of reads in the sample. And what that allows us to do is we can take these TPM values and we can compare them between genes and samples. That's something we cannot do with raw counts. And the reason for that is, if I wanted to say something like, or if I wanted to ask the question of, is gene 1000 here expressed at a higher level than gene 100? If you look at the raw counts, for the controls, for example, here, you might say, yeah, it looks like there is about a 10-fold increase in expression there, right? We're averaging about 20 here with gene 100 and 1,000 with, we're averaging 200 with gene 1,000 here, right? That's what it would look like from the counts, but that might actually be incorrect. It might be misleading, because the fact of the matter might be that gene 1000 here might actually have a transcript that's 10 times as long as gene 100. And if that's the case, then surely you would expect to get 10 times as many reads for gene 1000, since it's got a transcript that's 10 times longer, than gene 100. Okay? So we can't look at raw counts and compare genes, or gene expression levels, that way. No, instead, we have to normalize for the length of the transcript for each of these genes. And you can see here, that's the first thing we do when we calculate TPM, is we divide the total reads mapped to a gene divided by its length, or the length of its transcript. Once we do that, then we normalize that A value there according to the total number of reads that was obtained for that sample. And what that allows us to do is compare genes between samples. So let's say, for example, that I'm comparing this gene 100 again in the control samples to the transfected samples. Without normalizing for the total number of reads, or the total number of counts in this case, I can't really draw any conclusions about these numbers. You might look at them initially and say, okay, so looks like the average here in the control samples is about 20, and it's about the same over here for the transfected samples, so maybe there's really no change in expression for this gene between the two samples. However, remember, when we do mRNA sequencing, we're getting millions of reads, and the total number of reads that we get for a given sample could definitely change between two samples. So, in other words, what if, let's say, for these control samples, I only got about 10 million reads for each of them, but then I got 40 million reads for each of these transfected samples. That would be the total number of reads I'm getting here. Then that would mean that this average of 20, right, would be a larger fraction of 10 million reads than an average of 20 would be over here for a total of 40 million reads for those samples. Okay? So even though the counts might be similar, the percentage of the total reads for that sample might be different. So that's why in the second step of calculating TPM here, we take the individual A value from before and we divide it by the total number of reads that we got for that sample. And then we have a correction factor here of 1 million just to make things make a little bit more sense, because there's millions of reads, so we are going to take that sum there down by a factor of 1 million. Okay? So we can get these TPM values that allow us to compare expression values. The DEC2, the DEG analysis, told us which genes were significantly different between the samples, but the TPMs tells us the magnitude at which they were expressed. Okay? And this can be very important because you might very well run the DEC2 analysis and find that, let's pick an example here, gene 3433. It might be significantly different between the two samples. It's got the low p-adjusted value, and it might have a large log two-fold change here. See that -5, that's 2 raised to the 5th power, so that's a 32-fold difference between the control and the transfected samples. However, if that 32-fold difference is between a TPM of, let's say, 0.1 for the control and 0.32, or no, that would be 3.2 for the transfected sample, then that's still a gene that's not expressed at a very high level at all in those cells. So we might get a little bit less excited about that type of DEG because the TPMs are so low. Okay? So whenever you're trying to identify genes that might be involved in a certain response, it would be a very good idea to consider both the data from DEC2 that tells you the p-adjusted and the log two-fold change values, along with the TPM. And what we would want there is a DEG that does have that significance factor, right? P less than 0.05, but it should also have a TPM that's expressed, at a meaningly high level. So something greater than 10 usually is what we say. Although that's definitely an arbitrary threshold that changes between users and experiments. Okay? But there you go. These are all the wonderful things that Galaxy can give us. Okay? And so without any further ado, let's jump right into it and go over to Galaxy here. All right? And you can also download the protocol here, or it's part of the lab manual that's posted to Blackboard. Okay? That's going to talk you through all of the steps that I'm about to go over. All right, so you can see here the first step is to go to the website and create a username and password so you can log in and they can save your data. All right, so you go into here and that's what this should look like once you're logged in. And this is where you should start. The first thing I would do is I'd come over here and on the right side menu there's what's called a history. This is where we're going to keep track of all the files that you import or that you generate, okay? And sometimes you'll have multiple histories for different projects, so I'd suggest that you start by labeling this something that is meaningful for this project. So something like PC3 would probably be a good idea, okay? So we've got our history labeled there. The next thing we need to do, though, is we need to import our mRNA sequencing data. And there's a lot of different ways you can do that, but what you do is you come up here to this button, Upload Data, and what you can do here is you could choose local files. So if you had the fastq files that we're starting off with here on your computer, you could upload them by clicking this button. But I'm not going to have you do that in this case. Instead, we are going to fetch the data from a different source. Okay, so I'm going to click on this button And there we go. That brings up a field where I can put accession numbers here for a database that's called NCBI SRA. Okay? And that's where we've already uploaded the data that we're going to be looking at in this exercise. Okay? So what you're looking at here would be mRNA sequencing from some untransfected PC3 cells, along with mRNA sequencing data from some transfected PC3 cells. Okay, so we've got both types of sample in triplicate, and our fastq files already exist online here at this SRA depository. Let me see if I can click on that. Yep, here we go. And so here they are online. If you wanted any more information on them, you can see here each read is 100 bases long. All right, and we've got paired in data here. That's 2 reads per spot. And if you wanted to see, you could click on any of these links here, give you a little bit more information that we've uploaded about this study. All right. Also, depending on the size of the file, yeah, you can't do it here. Sometimes in this browser, you could even search the data to find specific sequences in these reads. But yeah, this file's too big. It won't let us do that. Okay. So anyways, this is where the data is coming from. It's freely available online. All right, but we don't even need to look at that right now. All we need to do is copy each of these accession numbers for those different files, and then we're going to paste them here into this field. Now, I want you to be very careful. You see there it added a 7th line to this file. I'm going to delete that, and I'm going to scroll up to make sure that there's no other extra spaces or lines or characters. Okay, because we have to be very stringent about what we're actually telling Galaxy to retrieve here. If there was an extraneous A or a space, that might screw up this entire process, and it takes hours to import this data. All right, so we've got that finished. Now we're going to click Start, and we're going to close that. And you should see over here in your history that we have a pasted entry. All right, so that's the first item in our history here. And it should finish processing pretty quickly here. I'd expect. Yep, there we go. And you can click on this, and there's exactly what you put in there. And if you wanted to, you could change the name on that, but we're just going to move on for now. Okay. So the next thing we need to do is actually import that data. So we told Galaxy what the name of that data is. Now we need to import it. So if we go back here to the protocol, we need to find this tool, faster download and extract reads in fastq. So over here on the left side menu, there's a lot of tools that are already built into Galaxy. It's a really fantastic resource that a lot of people have sacrificed their time to make it what it is. So we should all be highly appreciative of this. But anyways, you can click over here on Get Data. And yep, there it is. Faster download and extract reads in fastq. That's exactly what we want. So I'm going to left click on that. And then it brings up the tool in the middle here. For input type, I'm going to start by saying I have a list of SRA accessions. Remember, SRA is the name of that database, that depository that we just looked at where our files are. So we have a list of SRA accessions, and we have 1 per line. And then you can see here, Galaxy already guessed, well, you've only got one thing in your history, so it must be that. That's your accession list, which is exactly correct. So we've got that going there. And let's see, do we want to do anything else? No, I think that's it. All right, and then we're just going to click Execute. All right, so right now is a very good time to pause this video, go have a meal, maybe go sleep for the night, because this process, depending on when you're doing it during the day, how busy the server is probably going to take several hours to complete. It would be a little bit shorter if you were uploading this data from your personal computer, but unfortunately this is as fast as this wonderful free software goes. So yeah, this will take a little bit of time. So go ahead and pause. Yeah, here we go. So you see it's just starting to go and retrieve that. All right. But yeah, go ahead and pause this and then resume the video whenever all of these files are here. Okay? But me, myself, I'm going to do a little bit of time travel here. I've already uploaded these data to another history. So I can come up here to view all histories. And here we go. There's my other PC3 history. And I'm going to switch to that. All right. And then I'm going to click this home button to go back to Galaxy. There we go. Okay, so please ignore all of this other stuff that I've already done. But once your instance of a Galaxy has finished retrieving that data, it should look something like this. So there's our pasted entry, but now these other 4 items that were created by the get fastq tool should all be green. All right, so there's a lot here. There's a log that if anything went wrong, it'll tell you exactly what went wrong. So maybe you typed in your accession number wrong, it would probably divine that for you there. But what we're interested in is this entry right here, this single in data. Now, this might be a little confusing at first because if you recall, I told you we were working with paired in data in this exercise. And indeed we are. So you might think it should appear here under pair in data, but if we click on that, it says this list is empty. That's because whenever this specific tool in Galaxy imports the data from SRA, it takes both of the paired in reads and it merges them together into a single file that it just by default names whatever the accession number is. Okay, so yeah, sorry for the confusion here, but you are looking at paired in data that's been combined into a single in data format. All right, and if we click on this, we can see exactly that. So here's one of the first reads from a spot, or here's the forward read from a spot, and if you go down, you'd see the second read from that same spot. Okay, so it's combined both of those files, which were about 2 gigabytes, into something that's 4 gigabytes large. All right, but there you go. That is exactly what we want. Now, all of our fastq files are uploaded to Galaxy, so we can continue on to the next step. All right, and so if you're following along in the protocol, here we go. The next thing we're going to do is use HiSat2 to generate that BAM file, which is the alignment of all these reads to the genome. Real quickly, before I move on, I do want to just acknowledge that everything I'm about to say or show you has been modified from this tutorial here. That is online if you want to click that. Galaxy has a lot of training tutorials that are available online. So if you're ever hitting a wall, if you ever can't figure out how to do something, or if you'd like to try something new, I'd encourage you to check out their training archives because they have everything you could ever need. And so I've distilled one of their tutorials down to exactly what we need to do in our exercise. But if you want more information on this exercise, you can go to that website right there and they'll give you more info. Okay? So anyways, this is the next tool we need to use, this HiSat2. So I'm going to go back here to Galaxy. I'm going to go to the left menu, and there's this search bar here. And I'm going to type in HiSat2. And then within a few seconds, yep, there we go. You can see there's HiSat2. I can click that, and it brings up that tool in the middle. Now here, there's a few more fields to fill in, but it's still not very bad. First of all, since this is an alignment tool, we need to tell it what we're aligning to. And in this case, we're going to use a built-in genome. This is a genome that's already present. It's part of the HiSat2 tool, but you could definitely import a genome as well into your history. So let's say you were doing mRNA sequencing on a new organism and the genome has just been sequenced. You're the only one that has it. You could import it here into Galaxy, and you could still use HiSat2 to map your reads to that genome. But you would select this option. We're mapping to the human genome, which is very well known. And so it's already built into the HiSat2 tool, and that's the option we're going to select there. Then we're going to come down here to reference genome and just type in Homo sapiens. And here we have an important choice. You can see there's actually a few different choices for what I want to use here. All right, but the main differentiator between all of these is this HG19 versus HG38. These are actually different drafts or different builds of the same human genome. But the larger the number, the more recent it is. So this HG38 is the newer draft of the human genome. And so that's the one that we want to use because there's some errors, there's some other problems with HG19 that have been revised and fixed in HG38. Okay? Now, if you wanted to go into higher resolution here, you could also pick something like HG38 canonical female. That's going to assume that the genome has 2 X chromosomes and no Y chromosome. All right, and maybe you need to do that for one reason or another. Maybe your specific project is sex specific, but ours is not. So I'm just going to click HG38 here. So now HiSat2 knows which genome we're trying to align to. The next question it's going to ask is this a single or paired library? And here again, we have to be careful. Our fastq files were paired end, but the tool that we use to import them combined them into a paired in data from a single interleaved data set. So interleaved means that we combined both of our paired in files into single files. So that's what we need to select for this specific exercise. If you uploaded that data yourself, those data would probably still appear as 2 different files, and you'd have to select paired end data there, not interleaved. Okay. Once we do that, then it gives us this option to choose which set of fastq files we're talking about. And so for that, we're going to click here, Dataset Collection. And then in this drop-down menu, you can see we've got some choices. So do we want to look at the collection that's pair-in data from fastq dump, single-in data, or other data? We want this one, single-in data, because remember, if you look over here on your right side menu, that's where it dumped all of our interleaved paired-in data, is in that single-in data collection. Okay, so that's what we want to select here. Sorry again for that confusion, but our paired-in data ended up as single-in data, and we're looking at that collection there, the one with 6 items in it. Okay. Now down here it says specify strand information. Now this again is something that you should know about your data, either because you did the sequencing yourself or the sequencing company told you. We did a library prep that was unstranded or stranded. Unstranded is the more common way to go about this. That just means that any one of your reads could come from the sense or the antisense strand of the human genome. Okay, there are some methods that you can use to select for just one strand, but we didn't do that here. If you don't know if your sequences are stranded or unstranded, there is a way to check that, but you can only do that after you've done this alignment. All right, so you could circle back around to do that. I'll briefly show you how to do that later. All right, and then let's see, am I forgetting anything? I think that's it. Okay, so then we click execute, and this is exactly what you should see. We're using all of the data in that single-in data collection, and it's going to generate 6 different files for us. So remember, we had 6 samples and 6 of those fastq files. So we're going to convert every one of those fastqs into a BAM file. All right, and once again, you should have something on your history now that looks like this. It says HiSat2 on collection 3, and it gives you a nice little status bar here too. But the unnice thing about that is that, again, this alignment is going to take probably a few hours. It's generally about 30 minutes per file. So yeah, here you might want to pause the video and come back whenever that specific item has turned green once you have those BAM files. Me, again, I'm going to travel through time. Actually, before I do that, I'm going to delete that job. Yeah, there we go. Okay, so I deleted that, and then I'm going to come down here. There we go. This is what you should see whenever that is done running, right? So this is turned green, and I can click on that, and it's giving me 6 BAM files. So we can see the designation there, BAM, and they're all named the same exact thing that they were as fastq. And I can click on that again, and then I can scroll down here, and there's actually a few cool things that you can do at this point. So once you have this alignment, you can click on one of these links here. I like this BAM IOBio link, for example, and it'll actually show you your data. So what you're looking at here is where your reads aligned to the genome, right? So here's all your different chromosomes. You can zoom in on chromosome 1 and it shows you, all right, so you've got a lot of reads there. This isn't all of your reads, but you can see that there's a pretty good distribution, right? And over here, this pie chart is telling you that 98% of your reads were mapped, so that's good. A few were discarded, probably due to low quality matches. If you remember when we were setting up HiSat2, you had to tell the program whether you had stranded or unstranded data. Here, this metric right here is telling us that 51% of the reads map to the forward strand of the genome, and then 48.8% map to the reverse strand. So this is what unstranded data look like. You have an equal probability that you're sequencing from the forward or the reverse strand. If this was 100%, then they would be stranded. But this is unstranded data. All right. And let's see, what else can we tell here? The mapping quality is pretty good. So that's nice to see. But another thing we can do is let's look over here at the sex chromosomes. So for example, you can see a lot of reads mapped to the X chromosome. But then if we look at the Y chromosome, there's not a lot of reads. And the few reads that we have, if we look down here at the quality, is actually pretty low. Our qualities were up around 60 in that other chromosome. Now we're down around 2. So we've got just one low quality read here. And so what that's suggesting to me is that these must be of a female origin, the cells that were used to generate this data. So yeah, I think that's kind of neat. And I'm just showing you the 10,000 foot view here. You can zoom in a lot more than this if you want to, and you can actually see how many reads were mapped to each gene on these chromosomes and where they mapped. So there you go. I show you this because it really does a good job of illustrating what we just did. HiSat2 took all those random sequence reads and it actually mapped them and showing you where they correlate or where they align to in the human genome. Okay. So good. Now we've got our BAM files, and look at that. All of these are 4 gigabytes large. So we're going to fill up that Galaxy quota pretty quickly. I've got some other things in here, so don't be scared. You've probably got a lot more room than I do. But there we go. We've got our BAM files. Okay. The next thing we need to do is we need to take those BAM files. Remember, those are just alignments. And we need to measure the counts per gene with another tool called feature counts. So once again, I'm going to come in here to Galaxy's menu, and I'm going to type in feature counts, no space. And there it is. So here's what the feature counts tool looks like. The first thing I need to do is tell it which BAM files we're going to be looking at. So again, I'm going to click on Dataset Collection. All right, and you see it changed over pretty rapidly. It figured out, all right, you've only got one set of BAM files, so that's what we need to be looking at. Again, specify strand information. We are unstranded. It's also, though, going to need to have some sort of file that annotates this reference genome to indicate where the genes are. All right? Because if you come back here, remember when we're talking about BAM files, all they do is give a position. Ab initio, there's no way Galaxy can tell if that position is inside a gene or not, let alone which gene that might correspond to. Okay? So you have to have some sort of gene annotation file that you can use for this feature counts tool. And again, if you had a novel organism, you may have made that annotation file yourself. You could upload it here to Galaxy and use it for that purpose. Or, if you're lucky like we are, in this exercise, you can just use one of the built-in annotation files. So the one that we're going to use, again, is that HG38. You can see what happens here is feature counts already guesses that since we used HiSat2 with HG38, we're going to want to use HG38 here as well as the gene annotation file. Okay, but I do want to point out that there are also built-in annotations for mouse genome as well, MM10 and MM9. You could also use here if you had murine cells that you were trying to analyze. All right, and then if we scroll down here, it also lets us choose an output format. We're going to use the default because the next tool we're going to use is that DESeq2, but I do want to point out that you could output a file here that gave you read counts and gene lengths. And that's important because going back to our discussion on TPM, remember, you need the length of each of these genes to calculate a TPM. So if you wanted to do that, calculate TPMs, you would need to know the length of those transcripts. Now, in our next video, I'm actually going to give you a worksheet that already has these gene lengths in it, so you don't need to retrieve them here. But if you ever needed that info, that's how you would get it. Okay, so we're just going to select this option here. All right, and then let's see, is there anything else I'm forgetting? Oh, yes, options for paired end reads. So we do need to click this link right here, and we need to enable it to count fragments instead of reads. Okay, so that's just a unique thing that we do for paired end reads. And then we're ready to click execute. So feature counts isn't nearly as bad as the fastq import or the HiSat process. It should only take about 30 minutes. But once again, go ahead and pause the video, go do some other homework, and then come back to me. But again, I'm going to travel through time here. And oops, I'm going to pause those jobs so Galaxy isn't unnecessarily burdened. But here we go. This is what you should end up with here. Okay, some sort of feature counts data. And you can see here's one where I did it with the length incorporated. So this file is going to have a few things. First of all, it's going to have the gene ID, so the numerical identifier for the different genes. And then it's going to have the counts here in the middle column. So this is the counts, the reads that we saw for every gene. But it's also going to have the length of that given gene. Okay? So this is what you would need if you were going to calculate TPMs next. But that's not what we're going to do. In this video, I just want to skip that step and go straight ahead to DESeq2. Okay? So in that case, it's actually not that file that I want. It's, let's see here. I ran it again. There we go. Your data, your file should look like this. So it does still have the gene IDs, it still has the counts, but you can see the lengths are missing here. And DESeq2 just wouldn't know what to do with the lengths, so that's why we omit them. It's not that it would, necessarily be bad to have, but DESeq2 is only looking for 2 columns in this data. Okay, so there we go. We've got our lists of counts, and now we're able to go on to the next step, with DE-seq2. All right, so let's just scroll down here in the protocol. Here's where we are. Oh, sorry, I wanted to do one thing before we move on, which is download the count data. So this is just a good spot to save what you've done so far, because you certainly don't want to lose all those hours of processing power if, let's say, for some reason you delete this history accidentally or lose power or a server explodes somewhere, okay? So anyways, what we can do next is we can take these individual files, okay, that we have for each of our alignments, each of our samples, and we can come over here and use a tool called column join. For some reason, it always comes out down here. Seems to me like it should be the first result. Anyways, we can come in here and we can go to our dataset collections, and which one was it that we wanted? It was the one without length here. Let's just say this one. Okay. So we want that one right there. We want to take all of those individual files and staple them together into a single Excel sheet, or sorry, not an Excel sheet, but a CSV or a TSV file, a text file that we can download from Galaxy to save to our computer. All right, so I'm going to select that file there, and then for identifier column, I'm going to put in 1, number of header lines in each input file, put in 1 again. This is just telling column join that there is going to be a top row that tells me what those data are, okay, and we're going to keep that. And then I'm going to scroll down here and click execute. Okay, and this should go pretty fast. We shouldn't need much time at all. Let's see, has it already run down here? I may have deleted that other one. Let's give it a second. There we go. So we click on this, and then now you see we still have the gene ID in the first column, but now all 6 of our other reads columns have been joined to that. And I can click this download icon. I can save that to my computer here. All right, so let's just call this counts. And then I can go into Excel. Let's get a new sheet here. Or actually, no. Let's use this. Oops. Yeah, let's go to a new sheet. All right. Blank workbook. And I can come in here to data, import from text or CSV. And then I just have to navigate to where we saved that file. I believe I saved it here in the class file. And let's look at all files. That was counts, right? Yeah, here we go. So there's counts. Click on that. The import wizard should power up here. Please be patient with it though. There's going to be about 30,000 rows of data here. And we'll load that. And there you go. So now we have all of the counts for our different samples. And right now the fastq names are up there, but I believe this was our first control. This was the second one, third one, and then these were our transfected cells. So you can annotate that, you can go through here, you can sort that however you like. But just as a reminder here real quick, when you save this, you want to make sure that you save it as an Excel file, okay? Not as a text file, for example, which is what it might ask you to do. So there we go, we're saving it as an Excel workbook, save that as counts, and there we go. Now we have our counts file. But remember, we can't really do much with this because the data need to be normalized. They need to be analyzed for with statistical methods to see if there is any significant differences between, let's say, this triplicate of control cells and this triplicate of transfected cells. So I'm going to get rid of this file for now, but it's good that we've saved that on our local machine, so now that data is safe. Okay, so we'll revisit that data in the next video and calculate those TPMs. But for now, let's go on to DESeq2. So I'm going to go back into Galaxy, type in DESeq2, and there we go, there's the tool. And now we're ready to run those comparisons. Okay, so the default here for how are we going to compare them, we're going to select datasets per level. That's fine, that's what I want to stick with. And so the first thing I'm going to need to do is name a factor. So this is the experimental variable that we're testing in this experiment. So I'm just going to call this something like transfection effects. Okay, so what is the effect of transfection on these cells? And then I'm going to come down here, and you see by default it gives me two different types of samples that I'm testing here. That's what these factor levels are. Okay, so I can put in something like these are my transfected cells. Okay, and then I can come down here to multiple data sets, click that, and what I want to do in this field is I want to put in the fastq files, or so not the fastq files, but the counts for each of our transfected samples. Okay, so first off, let's clean up this field. If you have anything in here, you can always delete it by coming over here to the history, and you can see like this column join. I don't need that anymore, so I'm going to delete that, and it disappears from here. Same thing with this DESeq2 result file. That's from when I ran DESeq2 previously. Okay. All right. So now I can come in here, and this can be a little bit messy. One thing I was going to tell you to do was it's usually a good idea to rename these files as you make them. So if we come over here, like with this feature counts, we can click this edit attributes button, and I could rename that. This is control 1, for example. The next one is control 2 and control 3. That's probably a pretty good idea to name your specific files. So when you get into this situation here with DESeq2, let's go back to it, then it's easier to tell what you're actually doing. Okay. Oh, but I need to put back transfection effects and transfected. There we go. But I can tell you right now that the way we've done this, remember the first files that we put in were the untransfected files. So those are going to be the first ones that pop up here with feature counts. So this item 36, 38, and 40, those would be the counts from the untransfected samples. The files in between here, these are summaries of the feature count analysis. That's not at all what we want. We want the counts here, and these are the controls, the untransfected samples. So if I'm putting in transfected samples first, that's going to be the next three. It's going to be this one, and this one. Okay? And if you're doing this at home, your labels here should look pretty much the same, unless you've renamed them. Okay? All right, so I'm going to click all three of those, then click OK, and you see they pop up here. So these are my transfected data sets. Now I'm going to come down here to factor level 2, and I'm going to say these are my controls, or untransfected cells. You can call them whatever you like, but you're going to come down here to browse data sets again, and now I do want those controls, so I'm going to click these 3 samples. Okay, not the 3 up here that I did before. So we've got items 36, 38, and 40. Those are my controls. 42, 44, and 46 are my transfected cells. All right. And then there's a lot more that you can adjust here with DE-Seq2. Now, the intention of this overview, or this video, is just to be an overview of how these tools work, so I'm not going to go into detail as to what each one of these things mean. But what I am going to say is these default choices are pretty reliable. You probably don't need to change too many of them for most applications, but it is the kind of thing where I would encourage you, if you're going to be doing a lot of mRNA sequencing analysis, if this is going to be a large project for you or even a career, please look into more on how this tool works. For now, we're just going to gloss over all of these different variables. Okay, scroll all the way down, and here we go. I think that's everything we wanted to do. Let me just check my notes. Yep. All right, and then we're going to click execute. And good. So we're using the 6 inputs, which would be the counts files from the untransfected and the transfected files. Okay. And what this is going to do for us is it's going to give us a results file that's going to give us that p-adjusted value and the log2 fold change between the two different types of data sets for each gene. Okay. There's also going to be some plots that it shows us as well. Okay. So this analysis shouldn't take too long. I'm just going to let the video run here, but it really should only take a few minutes depending on how busy the server is. But feel free to pause the video and we'll come right back to it. Okay, so here we go. DE-Seq2 is done running. And let's first take a look at the plots that it prepares. All right, so these are all available in the form of a PDF. You can scroll down here and download that. All right, let's get that. Open it up. All right, so in here is going to be some useful little references, okay? So for example, you can see these comparisons of gene expression between the different samples, but what I'm most interested in would be this, the transfected versus controls analysis. So you can see there's quite a few different genes shown in red here that were significantly upregulated in the transfected cells versus the controls. So those are the red circles that are above this baseline. There's also a few that were downregulated as well. So this is telling me that, wow, I do actually have something to look at here. All right, but I'm going to close that for now, collapse that, and then come in here to the DE-Seq2 result file. All right, so again, we're going to scroll down here. And this is going to give us a tabular file. I'm going to save that as well here. DE-Seq2, I'm just going to call that real quickly. All right. And we can import that into Excel. All right, data again from text CSV. Okay, and where are we? Classes, all files, and scroll down to DE-Seq2. Again, this is going to be a large file, so please be patient with Excel. Load that up. And oh, it didn't copy the headers. Well, let's put that info in here. So what this is showing you from left to right is you've got the gene ID over here. I believe it's then the base mean. Then it goes the log2 fold change. And what are the other headers? Yeah, base mean, log2 fold change. Standard error, some statistical info there, standard error, walled stats, p-value, and p-adjusted. There we go. Okay, so now we know what everything is. So let's walk through this. Here are the data that we really want. We want to know that log 2 fold change. We also want to know this p-adjusted value over here. Okay, so you may have noticed that there's two types of p-values that are generated with this DESeq2 output. There's the p and the p-adjusted. So with the p-adjusted, what we have here is a p-value that's been corrected via the Bonferroni method. All right, and what that pretty much does is it takes into consideration that DESeq2 here is looking at a very, very large number of samples. Okay, so when we're considering which differences are significant or not, as that type of analysis tends toward infinity, you might have some things that appear to be significantly different because the sample size is so large, but they're actually not. And so just in a nutshell, that's the correction that we're using here with the p-adjustment. So this is a more stringent analysis of statistical significance here, and that's what we want to use, okay? So with that being said, it's the p-adjusted, not the p-value that we're looking at, and we want this log 2 fold change, okay? So let's go ahead and identify some DEGs, some differentially expressed genes here. So the first thing I'm going to do is I'm going to take all of this data. So I'm going to highlight those columns there, then press Control Shift and down. Oops. Let's do that again. Highlight my columns, Control Shift down, and that's going to highlight all of my data. There we go. And then I'm going to go over here to Data, Sort by column 7. That's where my p-adjusted values are. And I'm going to do smallest to largest. There we go. All right, so here we have our smallest p-values. And remember, the reason that we have p-adjusted values is we're trying to determine which differences are significant or not. And so a low p-value indicates that it is, a higher value indicates that it's not. Specifically here, we're defining that threshold as p less than 0.05, indicating significance. So these are really small numbers. I'm going to go down quite a ways here. Here we go. Almost there. Right about there. There it is. So you can see here, after we go through 1,800 genes, right there, I'm going to insert a row, because every one of these genes down here, since that p-adjusted value is greater than 0.05, these we can define as not being significantly higher or lower in the transfected cells compared to the untransfected cells. Okay? So these are genes that are not upregulated or downregulated. But you can see here we still have 1,800 different genes to choose from. And that's a rather long list, right? So I don't want to be overwhelmed by that. So that's why when we define a DEG, it's not only as a gene that has a p-adjusted less than some threshold value like 0.05, but we also look for a log 2 fold change value that is greater than 1, or another way to say that for a downregulated gene would be less than -1, because that means it has been changed 2 to the 1, which would be at least 2-fold between the different samples. Okay, so what I'm going to do now is I'm going to take this data here. Again, I'm going to highlight those columns, hit Control Shift and down. So notice it stopped me there at that row that I inserted. So now I've only highlighted the genes that are significantly different between the samples. All right. And then I'm going to do a data sort, this time on column 3. All right. And we're going to go, in this case, largest to smallest. All right. Oh, good. I got my headers back. Accidentally lost those. All right. So what you can see here is that my p-adjusted values are still low, as they should be. But now I've rank ordered everything by this log 2-fold change. So you look at this and you say, wow, this gene ID, whatever this is, 91543, that changed 2 to the 8th fold. So what is that, about 128-fold between the transfected and untransfected cells? It was upregulated. Anyways, let's scroll down here, and we see there's a lot that are in the same boat. But eventually, we're going to reach this cutoff point here. Here we go. Where that log 2-fold change is less than 1. So I'm going to insert another row there. But there we go. Now we've gone from 1,800 genes to only 600 genes that we're calling DEGs. These 600 genes have a p-value that's less than 0.05 and a log 2-fold change that's greater than 1. Okay? That's what we want to see there. So these are all our significantly upregulated genes. Now I can do the same kind of analysis. Let's see, let's go down here. For downregulated genes, I could highlight these data, Control-Shift down, do a data sort on them as well, but this time go from smallest to largest. Again, based on column 3, that's my log 2-fold change. There we go. And whoops, what happened here? Oh, I'm just down at the bottom. Okay. Oh, what did happen here? Did it change that entire column? Ugh. Well, anyways, this still shows what we're trying to get at. Is here now, we're looking at these genes that are significantly downregulated because that log 2-fold change is negative. And again, I can scroll down here to where we have that -1 threshold. Here we go. I can insert a line there. And now I can say about these genes, these 385 genes that are up from that row, they were significantly downregulated. Okay? These genes were downregulated too, but it was at less than 2-fold of a difference. So I'm not really confident that that small of a decrease, even though it's significant, I'm not confident that small of a decrease would have an effect on the cell. Okay? So those are the thresholds we use to define what our DEGs are. Okay? So you want to do that, and then you know what your differentially expressed genes are. All right? So I believe that's everything I wanted to do here. Yep. Okay. Yeah. So that's everything I wanted to do here because that shows you how to analyze all the data in Galaxy. The purpose of the next video will be to take a closer look at these DEGs using the TPM values and the log 2-fold change and the p-adjusted values to decide which of these genes might be the most important differences between these two sample sets, between the transfected and the untransfected cells. But all that work goes on in Excel, so we're going to kick that off to the next video. If you have any questions about any of this, please do let me know.